The bacteria that we are going to study about today is Corynebacterium. In this genus Corynebacterium, a very important species that we need to study about is Corynebacterium diphtheria. Corynebacterium diphtheria is a pathogenic bacterium that causes the disease diphtheria. This bacteria is also known as the Klebsiella bacillus because it was discovered in the year 1884 by a German bacteriologist, Edwin Klebs. And another another scientist was there whose name was Friedrich Loeffler. So both of them together discovered this bacteria, and hence another name given to it is Klebsiella bacillus. This is a rod-shaped bacteria, and hence this is considered to be a bacilli, okay, not a cocci. Now coming to the morphology of the bacteria, Corynebacteria are gram-positive, non-acid fast, non-motile rods with irregularly stained segments and sometimes granules. They frequently show club-shaped swelling, and hence the name Corynebacterium. The word Coryne has come from the word club. Club means a kind of swelling, you know. See this diagram. This picture, it shows the Corynebacterium. Can you see this black or darkly stained area of this bacteria? This area is called the club. Okay, this is what somewhat similar like a drumstick, and then the arrangement you see here is somewhat like a Chinese letter arrangement. Here, this arrangements you see, just observe, it looks like the Chinese letter arrangement. Okay, so this is why, because of the club shape that I mentioned just now, this bacteria is known as a club-shaped bacteria, and hence the name Corynebacteria been given to it. The most important member of this genus is Corynebacteria diphtheria species. That is the causative agent of diphtheria. So we are going to study about Corynebacterium diphtheria now. Humans are the sole pathogen reservoir for diphtheria. Infection sources include infected persons and carriers, which is very rare. So accordingly, humans are the only reservoir, or the only one kind of host who is the source of this bacteria, diphtheria. Okay, this infection may be spread from one infected person to another, and the source therefore is an infected person, or sometimes a person may be just a carrier but not infected. Okay, but that circumstance is very rare. The disease is usually transmitted by droplet infection, or less frequent indirectly via contaminated objects. It is an acute and contagious infection characterized by pseudomembranes of dead epithelial cells, white blood cells, red blood cells, and fibrin that form around the tonsils and back of the throat. So what happens when a person is infected with this bacteria? So this is a contagious infection. Okay, a person may easily spread this infection to a healthy individual, and when a person is infected with it, the characterization of this, how is it identified? This is identified when a person or a patient develops a pseudomembrane. Pseudomembrane means at the area around the tonsils and the back of the throat, there is development of a membrane, a false layer, which is actually not a part of the body, but it is developed. How? because of deposition of dead epithelial cells, white blood cells, red blood cells, and fibrin. Okay, so that is formed around that area, and that kind of membrane is called as pseudomembrane. It is an uncommon illness that tends to occur in unvaccinated individuals, especially in school-aged children, those in the developing countries. It may happen to the elderly people and the immunocompromised patients. The virulent and toxigenic strains are lysogenic and produce an exotoxin, which is formed by two polypeptide chain. Okay, so this species have certain strains which are virulent and they are very toxigenic. So this kind of species, or sorry, this kind of strains, they produce an exotoxin. Okay, they produce an exotoxin, and that is formed by what? By two polypeptide chains, which is itself produced when a bacteria is transformed by a gene from the beta prophage. We'll study that way. Now there are four subspecies of the diphtheria. Clostridium itself is a genus. Within the genus, the diphtheria is a species, and within the diphtheria species, you will find some subspecies. Okay, so there has been four subspecies that recognized so far. They are Clostridium diphtheria mitis. The next one is intermedius. Third one is gravis, and the fourth one is belfanti. The four subspecies differ slightly in their colonial morphology and biochemical properties, such as the ability to metabolize certain nutrients, but all may be toxigenic and therefore cause diphtheria, or they might be non-toxigenic also. Unusually, the diphtheria toxin gene is actually encoded by a bacteriophage, which is found in toxigenic strains, not on the bacterial chromosome itself. That means what they're trying to say here is that, first of all, yes, there are four different subspecies of diphtheria that has been recognized, and the name has been mentioned here. And this subspecies, if we grow them on the culture media, they will show slight differences in the colonial morphology and also in the biochemical properties. The biochemical properties, like how they metabolize certain nutrients when they are grown on the media. But all of these subspecies, they may be toxigenic. And if they are toxigenic, they will be responsible for causing diphtheria. A few may be non-toxigenic also. Also, it has been found that this diphtheria is probably having a toxin gene. Why are they confirming this, that they might have a toxin gene? That is because they have seen that a bacteriophage is a virus which infects bacteria. So they have seen a bacteriophage to be present on this diphtheria, and this bacteriophage is actually found only on the toxigenic strains. So if this bacteriophage virus is found only in toxigenic strain, then obviously there has to be a toxin gene in this diphtheria also, that due to which the bacteriophage is also seen to be encoding this particular gene, and this is not seen on the bacterial chromosome itself. So this is about the subspecies. Now if we move towards the clinical feature of the bacteria when of this disease, what kind of symptoms you see, or what are the problems that may happen in the person? First is respiratory problem. This bacteria actually targets the respiratory tract. So you would obviously find problems in the respiratory area following an incubation period of 2 to 4 days. Patients typically report upper respiratory tract symptoms. The symptoms are nasal discharge and sore throat. The posterior pharynx, that means the area behind the pharynx, and near the pillars near the tonsils, they are mostly involved in this. The onset is often sudden. That means the beginning of this infection is very sudden. Initially, you will find people suffering from a low-grade fever, then malaise and membrane development on one or both tonsils. And those membrane, it may extend towards the other parts of the respiratory system. Next, it also involves the cardiac area. Cardiac is the heart. The toxic effect in the myocardium characteristically occurs within 1 to 2 weeks following the onset of infection, often when the upper respiratory tract symptoms are improving. Okay, so now the symptom is changing. You see, initially it will affect the upper respiratory tract. Gradually, the symptoms of the upper respiratory tract improve, and as it starts improving, if the infection is not cured, you will find that the infection is moving onwards. Moving towards a different area. Now it's affecting the cardiac area. So there you will find this symptom occurring within 1 to 2 weeks following the onset of infection. But that time, the upper respiratory tract symptoms are improving. Manifestations are due to arrhythmias and congestive heart failure. Next is neurological. Then from cardiac, again, you see neurological problem also. Here the symptoms can occur immediately or after several weeks. Bulbar symptoms generally occur within the first 2 weeks after disease onset, and they can range from mild symptoms. That means you will find problem in swelling to bilateral symmetric paresis of the palatal and ocular muscles. That means you would find movement difficult, moving the palatal area, then the area nearby to your eyes, all these areas, movement becomes difficult. Okay, the bulbar symptoms may remit or progress to paralysis. Either it may reoccur again and again, or it may progress, and from those areas like palatal and ocular muscles, it will involve the other areas also, and finally it progresses towards paralysis, making the person difficult in moving. So paralysis of the proximal and the distal skeletal muscles over the next 30 to 90 days. Although recovery can be very slow, patients generally regain complete neurological function. Yeah. So yes, the recovery is slow, but it's not impossible. The people regain neurological function if they are treated properly. Secondary complications include aspiration from bulbar paralysis and bronchopneumonia from respiratory muscle dysfunction. The next problem is the skin. Even cutaneous infections occur often in more tropical climates, presenting as non-healing ulcers. Okay, in areas like the tropical regions, there you would find the person suffering from skin infections, and their ulcers occur, and those ulcers are non-healing. A recent surveillance study of Native Americans presenting to the Indian Health Service clinics in South Dakota recovered from the disease. How many? Out of 133 patients, only 6 people recovered. That is only 5% recovery was there. One of them had the skin ulcer. So this is the clinical feature of the disease. Now, the lab diagnosis. How are you going to diagnose this disease? To diagnose this disease, we need to first of all collect the sample. So, depending upon the part of infection, whether it's if the symptom seen is a respiratory tract infection, then you collect swabs from those areas. Then you can also collect swab from the lesions if it's cutaneous infection. So depending on the area of the infection, you will be collecting the samples. Once the samples are collected, then we are going to perform the gram staining and Albert staining. Okay, gram staining we perform for almost all the bacteria because that is a characteristic method. But specifically, if you want to identify and determine Corynebacterium diphtheria, you have to perform the Albert staining. The Albert staining is a process which help you to observe the metachromatic granules present in this bacteria. Okay. So when you perform the gram staining, what do you observe? That the bacilli is a slender rod with tendency to clubbing at one or both the ends. The bacilli are pleomorphic. That means you see different kinds of morphology. They are non-sporing, non-capsulated, and non-motile. They are gram-positive, but they tend to decolorize easily. The granules are composed of polymetaphosphate granules, which are more gram-positive from the rest of the bacteria. When you perform Albert stain, what will you see? You will see green color bacilli present with black color granules. Granules means, you know, small dot-like things, granular things, grain-like. So those are granules. Okay, so it's all about gram stain. And then now, if you perform a culture characteristic of this bacteria, then you'll find that the growth is very less when it's grown on ordinary media. Like you are growing it on a nutrient agar media, that's an ordinary media. So the growth will be very less, very scanty. But if you provide enrichment with blood, serum, or egg on those media, then the growth will be very good. The optimum temperature for growth is 37 degrees Celsius and the optimum pH is 7.2. It is an aerobic and facultative anaerobic bacteria. And for it, a specific media that you can use to identify it or cultivate it is the Loeffler's serum slope and telluride blood agar. Either of this media can be used for growing this bacteria. Diphtheria bacilli grow on Loeffler's serum slope very rapidly, and the colonies can be seen within 6 to 8 hours, which is long before other bacteria can grow. That means it's very fast. The growth is very fast on this media. Colonies are at first small, circular, white, opaque disc, but enlarge on continued incubation and may acquire a distinct yellow tint. Diphtheria bacilli ferment with the production of acid. Okay, but there is no gas formation. If we go for a determination of this, whether the fermentation bacilli can ferment, cause fermentation or not, you will see that it does ferment glucose, but then it only produces acid, but there is no production of gas. Similarly, it also ferments galactose, maltose, and dextrine. In dextrine, but then it does not ferment lactose, mannitol, or sucrose. Some strains of virulent diphtheria bacilli have been found to ferment sucrose also. It is necessary to use his serum water for testing sugar fermentation. Proteolytic activity is absent. They do not hydrolyze urea or form phosphatase. So these are the characteristics of this bacteria. Proteolytic activity, that means it causes lysis of proteins. That activity is absent. They do not know hydrolysis of urea or nor do they form phosphatase. Then about the toxins, what kind of toxins are here present in this bacteria? The virulent strains of Diphtheria bacilli produce a very powerful toxin. The toxigenic effect of the bacilli are due to the pathogenic effect of the bacilli are due to the toxin. Almost all strains of Gravis and intermedius are toxigenic, while only about 80 to 85% of the metis strains are so. Okay, so we have studied about the different types of strains of we have studied the subspecies of diphtheria. So we didn't do subspecies again. You will find different strains. Now, in the Gravis subspecies, almost all of the strain present within it are toxigenic, 95 to 99% toxigenic. Similar is seen in intermediates also, but in meatier strains, you will find it 80 to 85% of the bacteria are bacterial strain are toxigenic. Diphtheria toxin is a protein. Okay, it has two fragments, A and B. Both the fragments are necessary for the toxic effect. So when the bacterium it release the toxin is inactive and active on fragment A is mask. All the enzymatic activity of the toxin is present in fragment A, and fragment B is responsible for the binding of the toxin to the cell. So as I told you, this diphtheria toxin has got two parts, A and B, and both A and B, both of them have got their own duties to follow. The bacteria, in that toxin, the fragment B part does not have any virulence, or it does not have the toxicity or the toxic activity. The fragment B part is responsible only for the binding of the toxin to the cell, whereas the other part, that is fragment A, it has all the enzymatic activity of the toxin which is required for this toxicity. Okay, now next is the virulence test. How are you going to perform the virulence test? The virulence test, in order to perform that, you will have to have the diphtheria bacilli. That if the infection of diphtheria bacilli is to be tested for toxigenicity or for the virulence, so what can we do? We can do it by two methods. One is in vivo, another is in vitro. In vitro method means you performing the test outside the host, and if you're doing it in vivo means performing a test within a host. Okay, so here in vivo, if we talk about, then here it is done by a skin test, which is told as subcutaneous or intradermal test. Whereas the other one, that is the in vitro method, it is done on the tissue culture or by observing some precipitation. Okay, so in vivo, as I told you, is done through intradermally when you are injecting the toxins into the human being or the host. You say after injecting that, you will find there is production of antibodies, and that is determined by the reaction on the skin. That is the in vivo test. Now in vitro test is done by a precipitation test, which is called as LX gel precipitation test. What do they do in this test? A rectangular strip of filter paper. You take a filter paper, then the filter paper must be rectangular in its shape, and that filter paper is impregnated with the diphtheria antitoxin. Diphtheria antitoxin, how much amount? 1000 units per ml. That antitoxin is placed on the surface of a 20% normal horse serum agar in a petri dish. Okay, where this strip of filter paper is placed. Now, when the agar is set, the surface is dried and narrow streaks of the strain are made at the right angle to the filter paper strip. Then a positive and negative control is also put up on it. The plate is incubated at 37 degrees Celsius for 24 to 48 hours. The toxin that is produced by the bacterial growth will diffuse in the agar. Okay, it will diffuse means it will spread in the agar, and whichever area the antitoxin is present in that paper, in that filter paper, in that area, what will happen? The toxin will react with the antitoxin in whichever area there is maximum concentration of antitoxin, and that area, when they both react, they produce a line. That is a line of precipitation. That is a line, the antigen antibody reaction. Precipitation is one of an example of that. So That precipitation takes place in that area when that antigen and antibody both react to each other, and the presence of arrowhead lines of precipitates indicates that the strain is toxigenic. No precipitate will form in the case of non-toxigenic strains. If there is if you don't see any precipitation, that will be an indication that the strain is non-toxigenic. Similarly, for diagnosis, other tests like tissue culture test is performed, where the toxicity of the diphtheria bacilli can be demonstrated by incorporating the strains in the agar overlay of the cell culture monolayers. The toxin produced diffuses into the cells below and kills them. Then, what is the prophylaxis or how it can be prevented? Diphtheria can be controlled by immunization. What is immunization? Immunization means vaccination. So there are various vaccines that has been developed, like diphtheria toxoid, which is usually given in children. It's as a trivalent preparation. Trivalent because it contains 3 different vaccines. One is for tetanus, one is tetanus toxoid, and the another one is pertussis vaccine, and the third one is diphtheria. So these 3 are present in it, and hence it's a trivalent preparation, which is also called DPT or triple vaccine. This bacteria is sensitive to antibiotics such as penicillin, ampicillin, cephalosporin, quinolones, chloramphenicol, tetracycline, cefuroxime, and trimethoprim. Okay, so this is about the different aspects of Corynebacterium diphtheria.